Adrian, thank you so very much for joining us today. We are big fans of your work, of your
Oh,
YouTube
awesome.
videos, of your science, and it's an absolute honor to have you on. Would you be able to introduce yourself to everyone of, where you are today and what kind of science you do?
Yeah, sure. Thanks for having me on. This is exciting. I am an entomologist. Formerly, I used to be called a myrmecologist. I used to just study ants, hence the name of the YouTube channel Ant Lab. But these days, it's sort of a mishmash of behavioral science on all sorts of different insects and things that are close to insects. And we also do some social science on humans, too. But I am a research scientist at the North Carolina Museum of Natural Sciences and also at NC State University in North Carolina.
How did you begin your journey in bug sciences? Like, what was it that attracted you, and when was it that attracted you to insects?
Yeah. I feel like I don't have the traditional story in that, like, I wasn't really into bugs as a kid or anything like that. I kind of like only got into one college, a state university in Florida, and needed a job like kind of immediately to help like kind of support myself a little bit. And my bio 101 professor was Walter Schinkel, who's a famous, he's now retired, but a famous fire ant biologist. So I got started kind of washing dishes in a fire ant research lab. And then that was like kind of mind blowing because I had no idea like you could study ants for a living. Like that just seemed bizarre to me. But it was like really cool and interesting. So I did that for like 3.5 years, and then kind of figured out that like biology is really interesting, and it's might be worth a shot trying to make it into a career. So my next step was grad school at Arizona State, where I also studied ant behavior, and then postdoc jobs after that, and finally landed here in North Carolina. So it kind of was, wasn't a grand plan for me. Just kind of happened. Kind of pursued my interest in animals and behavior.
Did you get to work directly with the fire ants and get to do any submergement of the ants? Like we've seen, like even at technical institutes, they're studied right for their different shapes that they can make as they're like kind of the semi-solid. Did you get to work with them as well?
Oh yeah, for sure. I helped. When I worked in the research lab, I helped on fire ant projects, on harvester ant projects, on fungus gardening ant projects. And I kind of started working on my own a little bit with trapjaw ants at the time. I mean, Florida, is at Florida State, which is in Tallahassee, northern Florida, and it's a super biodiverse place. And so there's a lot, there's kind of the US's greatest hits of ants are all there. So there's so much to work on, and a lot of the people, the grad students and postdocs at the time, were doing kind of interesting natural history work on all sorts of species. So yeah, when we collected fire ants, we, the standard way of getting them is to go out in sort of the early part of the day where it's sunny and they're all up at the top, like in the dome that's above ground, and to like dig up the dome, put it in a bucket, and then bring it back to the lab and drip it for half a day, an hour, and then scoop them out with a ladle into a little thing and collect the whole colony that way. So yeah, it was pretty wild for sure.
It's kind of crazy when you first see it, right? That you're putting a whole colony just in the water, and then afterwards they've kind of clumped together. You're like, oh, yep, got them.
Yeah.
Did you have a favorite ant or insect during that time at Tallahassee where that inspired you to go to ASU to continue on ants?
Yeah, I mean, I feel like 2 years into discovering that you could do this and working there, and reading about ants and insects, I asked one of the postdocs at the time, who's now, I think, a professor, maybe department head at University of Central Florida, Josh Keene, was like, have you ever heard of these things called trapjaw ants? He's like, oh yeah, I mean, we have them here in the forest, like they're common. I was like, oh, really? Are you serious? And so from then, was going out and collecting those and working with them, and they're still my favorite ants. I mean, they're so cool. There's so many different adaptations and behaviors, and they're smaller size in a colony. So like max is maybe 1,000 ants, but mature is like a few hundred. And so you can watch all these dominance interactions inside the nest, and all these really cool behaviors that sort of dictate how colonies function in systems like that. Whereas if you scale it up to like things like fire ants or leafcutter ants, you're like hundreds of thousands, millions, and like looking at an individual isn't necessarily too informative in that situation, but in like smaller ant societies like trapjaw ants, like you can really like watch an individual and learn about how that fits into the collective sort of colony organization, and that stuff like really fascinated me. And so that's what kind of like pushed me out to grad school to work on some more of that.
Would you be able to comment on how one would set up an experiment like that? So I know what our community really thinks is cool is just like running experiments and like the experimental method of how you come up with it. So how does one even go about tracking an individual ant in the colony, right? Like for example, a trapjaw ant, is it that they look different based on their caste system, or is it that you paint them? And then what are you looking for when you're framing that question?
Yeah, it's paint marking mostly with trapjaw ants, but other people have like done things like put wires on their petioles and give them little colored belts so you can track them that way, because sometimes ants will groom off paint when you paint it like on the back of their abdomen or something like that. So with smaller size colonies like that, it's relatively easy to give individuals or a subset of individuals colored marks like that and watch it over time. And so you can do that and set up a nest as it was. And I would do things like separate out a small group of individuals and then from a nest and watch them establish their own sort of hierarchy, or a situation where orphan workers would become reproductive and then recombine those parts and see what happens and see how they sort it out. And doing those types of experiments actually got me into video making, because a lot of the research involved recording stuff, and then you'd go back and analyze the video for your data collection, so that you didn't have to do it like real time and you might miss something. You can go back. And so for almost all of my research, I was recording stuff and then looking at the footage later and using it for science, and that kind of got me into like video making, media making, to explain and showcase sort of my science and insects in general. And so that kind of kickstarted where I am today in terms of media making and science.
Was that your first foray into media making? It was just like, here, do this research question that you have to record a bunch of stuff for?
Yeah, I mean, sort of. I had an interest in filmmaking too, and I thought maybe going to college that I would enroll in film school. And so like Florida State has a really awesome film school. And so when I got accepted and like showed up, first day I was like, oh, maybe I'll join the film program. And then I figured out that like, oh no, you had to apply to this when you're 16 and have this like whole portfolio of stuff. You can't just walk in. And so I was like, oh, never mind, we're not doing that. Can't do that. So yeah, I was kind of interested in media in the first place. And then when I was a graduate student, I kind of secretly enrolled in a community college film school to learn video editing. Like at night, I was at Scottsdale Community College at the time, had a film school, and so I enrolled there and was like a community college undergrad at the same time as get a PhD grad student.
Wow.
Yeah.
Was that driven by your passion and your love of videography?
Yeah, partly like maybe we should like think about a plan B here too in the middle of grad school. So yeah, it was like both of that. But all the stuff I learned there was kind of like, it just, it was before when YouTube tutorials were like the thing, and now you could just learn it by, watching a bunch of YouTube videos, but that wasn't an option there. So it sort of opened the door and like, how do people edit video and use computers to do it? I mean, at the time we were like capturing from recorded videotape and like putting it onto a computer and editing. So yeah, that was really useful for both research purposes, but then it kind of kickstarted me in like, yeah, maybe I could just make my own media and tell my own stories about science. Like instead of having to convince institutions to make a video with me or whatever, like maybe I can just DIY it and kind of learn it on my own.
Yeah, it's kind of frustrating, right? Like where it's the outreach and research aren't necessarily connected, but your stuff is like very clearly like the field expert, and let me make these really cool videos and show people things that they haven't seen before. So do you think your path into the science kind of fueled that desire to really share the knowledge?
Yeah, I do science so that I can like see and interpret the world in a new way, and like see something that maybe, or notice something or describe something that maybe no one's paid attention to. And I think that's a really cool thing that science can do. And at the same time, like, my reasons for doing it are to have that experience, and then media making and storytelling is just sharing that experience and communicating that experience. So I try to make content that I'm the only person that could make, because I'm in the situation of being employed as a scientist with some, degree of expertise or access to tools even to like make these observations. And so, I make them for contributions to science at large, but I also make them, out of that core reason to like see something new. And I feel like it's a responsibility of me to like share that with people and to be able to present that in a way that other people can enjoy it and get something out of, just like I get something out of doing it.
I think it's incredible, and it really shows that you don't want to gatekeep. So it's like this really refreshing mindset of how to do science, which I think is really, really cool. So when you went to ASU for graduate school, what drove you to go there? What was the ant that you decided to work with? And what was your research question?
I wanted to go there because, when I started looking at trap jaw ants and saw you could like study individual behavior in the terms of like, how does individual interactions lead to like a colony level organization or cooperation or things like that. I was really into like that type of study. And there was one researcher who just got hired at Arizona State, Jürgen Liebig, who had some really awesome behavioral work on ants, of like dominance interactions, of a thing called worker policing, and really kind of behavioral work that went into like how do ant colonies work as colonies. And so I had an idea that like, oh, that's the type of research I really want to do. So I applied specifically to work with him and doing that type of research. And so I kind of sought out like, these are the studies that I like to read, and I think it would be really cool if I could do things like that. So I applied to work with the person who had that expertise and would also be interested in those types of things. And so I got to Arizona State, and I enrolled as a master's student and was sort of given a project in that realm with an ant that's now called Novamessor. But at the time, it was a Phoenigaster. Which is a kind of a local desert ant. That some work in the 80s had been done about policing or controlling worker reproduction in the colony. And so the handover was like, why don't you follow up some of this work from the 80s and like see what you can figure out?
Is that a difficult primer for it? Because it's right, they're not actively working on it, so it's like, here, take this older knowledge and expertise and you get to run with it. Like, it seems a little bit imposing.
Yeah, a little bit. But in the meantime, since that work was done, like new techniques and new kind of ideas about how these behaviors work had been sort of advanced. At the time, it was like there was between then and when I was there was an advancement of like understanding about how ants communicate via pheromones, via contact pheromones, waxes on their cuticles. So it was like, explain some of these things that we observed and looked at, 20 years ago in the context of like new ideas and new kind of research and findings on how some sort of the communication aspect of these behaviors work. So it was kind of like, applying newer methods to an older kind of briefly looked at system. And then, I started working in that, and then transitioned pretty quickly into, after a year of that, into a PhD program. I didn't end up getting a master's. I just went straight to a PhD and continued to work on that for, 5 years at Arizona State.
What was the big takeaway of your thesis? What do you think was the coolest finding?
Yeah, I worked specifically on like how those ants use pheromones to determine who's reproductive and who isn't, and like how to control worker reproduction in the colony, and how to distinguish who's a queen and who isn't, and so how they communicate within the nest to keep colony structure as a reproductive monopoly. And one of the coolest studies was something we published in Animal Behavior. It was this weird little thing where if a worker became reproductive and somehow didn't get, policed or prevented from her nestmate sisters, and was able to develop chemical signature similar to that of a queen, even though it was a worker. Queens would actually seek that individual out and physically attack her, and then spray a compound from a gland called the Dufour's gland onto that ant, and then the rest of the ants would respond to that by ripping that worker ant into pieces and just murdering it
Wow.
inside the. Yeah, it was really cool.
That is such a metal behavior. So
yeah.
So when the workers are undergoing this, odorant conversion, do they actually get activated ovaries? Like, could they start laying, or is it just like a status that they're trying to go to be fed or what have you?
Yeah, it's both. So in that system, workers can lay viable male destined eggs. So they can, they won't mate, but they can lay haploid eggs that develop into fully fledged and functional males.
Is that because it's one of those systems where they need a mating flight and we just aren't able to do it in a lab setting, or is it that there's something functionally unique and different about a worker versus a true queen in the system?
Yeah, there's something functionally different. The workers are incapable of mating. They don't have a sperm storage, spermatheca, so they can't mate and lay diploid eggs, but they can lay a viable male offspring.
So the benefit for them is to be passing on their genetics then into the colony and just sneak it in. We know how often something like that in nature even happens? Like, it's a very unique behavior, right, to try to take advantage of a colony. It seems like even more unique that it's dependent at some point on the queen to mark that individual as almost like like the sus, quote unquote, like in the video games, right? Like, you're the one who's in trouble. Like, what if she doesn't get that signal in time? Is there a point of no return, or is it just always up to the queen? It's like, now it's up to you to mark this individual.
Yeah, there's there's a few like ecological things that go into it. So like, if the queen dies in a natural setting, the colony won't re-queen itself. So there is a natural scenario where workers will develop into male producing individuals in a nest, and so they'll do that. It's part of the colony life cycle. That there's, after the queen dies, there's kind of a last ditch effort to reproduce, and workers do that.
And is that many workers, and does policing presumably goes away at that point because it's that last ditch effort to save the genetic information?
Maybe in the lab, that species they still form a reproductive monopoly where there's, in a totally queenless situation. It's not like they go from a single reproductive to 100 reproductive workers. There's usually like, in the lab, at least one individual that's laying eggs.
Interesting.
Yeah, it is kind of interesting. Yeah, that if I would still work on that system, I would probably follow that sort of aspect up. And then ecologically in nature, even though there's a single queen, a mature colony will have anywhere from like 1 to 5 distinct non-connected underground nest sites. So there will be like fully fledged, that look like colony nests, that are totally, their queen's not there, but there still is brood and workers in there, and they're not connected underground.
How do they know that they have a queen still and that they shouldn't become a pseudo queen? Like, how is that information between like the unconnected nest held?
Yeah, I don't think it's necessarily known in that species, but in other ants, it's with the transfer of queen laid brood. So it's a transfer of like eggs and larvae from the queen inhabited nest to the worker inhabited nest. That sort of signals to the rest of the colony that like, there is a reproductive queen still here. So it's probably something like that. I'm not sure if there's causal evidence for that, but that's the supposed mechanism that works.
Yeah, it's always amazing to hear about different ant species and how complicated their systems are, and like all the unique additional behaviors that they have that, like you said, like it'd be really cool to follow up a lot of these different trails. Like, if in the lab it holds in the wild too about the one worker that gets selected, like why does that one worker get selected to replace the queen and to lay drones? Like, what about that worker is special? Is there, like with some ant species, right, there's a dueling behavior? Are they doing that, or is there just the luck of the draw? Like, it's a really, really cool, complicated question.
Yeah. I don't know. I mean, my guess would be that it's not really a competition, and that it's like there are certain workers that are in a physiological state that allows them to sort of do that quickly and like successfully. And so it's just kind of a developmental lottery. Whenever the queen dies, like if you're at that sweet spot, like, oh, here's your chance, right? But if you're, older and you're in a advanced, different physiological state, like maybe the window's gone or something like that. I don't know.
Did you, I'm sure there were many other really exciting experiments. Could you give us just another one? Like that smell one was so exciting. I'd love to hear another example too.
Yeah, the first scientific paper I ever published I was out of need to like get these ants in the lab. So a lot of people who like keep ants and work with ants will collect, newly mated queens and grow a colony from that. I didn't have time for that because, some ants like don't reach a maturity stage until years as a colony. And so I needed like entire whole colonies in the like mature colonies out of the ground. And in Florida, that's really easy to do because it's like sandy soil and you can just go dig like a 7-foot pit and get in it and get all the ants out of the ground. Like that's what I did in Florida a lot. But where these ants live in southeastern Arizona, like it's impossible. Like it's like rocky soil. You need an excavator, and then like you'd be like have a huge excavator bucket just smashing a queen, and there's no chance you would get a whole colony out. So then the question was, well, is this even possible? How do I get these ants in a mature state, like in the lab? And then my friend Kevin remembered some research about some ants evacuating their nests in response to things, in response to like floods, in response to predators or something like that. And so we were out in the desert looking for our ants, and they are active like at dusk and dawn and into the night. And another species of ant that is active in that time are army ants. And so army ants raid other, social insects, and like steal their brood for food, right? And so there's one species of army ant that raids a lot above ground and will raid into these nests to these Novomester or Phoenixester nests. And so then we had the idea of like, oh, I wonder if we could just go collect some army ants and like fake a raid and make these ants come out of the ground and then collect them. And that worked.
Wow.
And so we could basically like suck up 50 army ants, dump them into the nest entrance of these Phoenixester, and then like 5 minutes later, you'd have like 3,000 ants on the ground, and you could just stand there with a vacuum and like suck them up. And then the queen would either run out of the colony or like be carried by the other ants.
So they would evacuate the queen too for you. So there's no digging. There's nothing you have to do. That is
all brood. Just get out as quick as you can is their response strategy. So yeah, it was pretty wild that we could induce this just by collecting the right species of army ant and dump them into the colony at the right time, and the colony thinks like, oh, we're being raided, we got to get out of here, and then fake them and get them out.
How did that feel when that worked? That must have been the coolest feeling, right? It seems like it's a luck of the draw, and it's like it worked.
Yeah. No, it's astonishing. It's like again, it's like you'll get. I mean, these are the Novomester are big ants, and the Niva myrmex, the army ants, are like I don't know, 2 millimeters, maybe, like they're tiny. And yeah, you can get 3,000 of these Novomester just out of the ground in like 3 minutes. And they're just, everybody's just running as fast as they can out of the nest. And yeah, it's really cool. It's kind of like feels like a magic trick when you're doing it.
It really, it sounds like a magic trick, true.
Ants in like some panic state to get them. It's like you're like an ant whisperer out there.
It's totally what it sounds like.
Yeah.
When you collected the ants and you're bringing them back to the lab, given how like unique of an ecological niche that they have in this like dry soil that's difficult to dig through, like where did you keep them in a lab? Is it their typical like plaster and you dug up like some tunnels in it, or like was it something special that you had to do for them?
No, it was plaster, and we just kept them in kind of a humid environment in the lab, and had a big plaster nest connected to a different foraging arena where we give them food and everything like that. and they seem to do pretty well. We could keep mature colonies for years, and who knows how long they've been mature in the field before then. So yeah, it wasn't. They were particularly kind of easy ants to keep. I mean, some ants I've worked with, and bring in the lab, and they just do really poorly because they just kind of, I don't know, the conditions aren't right, and then like a sister species of them will live for years and years in the lab underneath the same condition, and it's just, you just kind of never know. And that goes into like some of the practical considerations for how to do this type of science, and what to work with and what you can't work with. And
we had a question from the chat earlier, kind of on that topic of, you mentioned that your favorites were the trapjaw ants. Did you have a least favorite?
Least favorite ants. I don't know about ant, but my least favorite thing I've ever worked with or filmed are adult cat fleas. That was bad. Like that, most of the things that you see on YouTube, I film here in the lab, and I don't film in a cage. And like they're flying, and they're like in a room, and then for every one video clip, there's like 2 minutes of me crawling on the ground, or like up like on the ceiling trying to catch the thing. And like I've only filmed 2 things in a cage really, like where they couldn't fly out and just at me. And one was a bald-faced hornet, and another was the cat fleas. But the cat fleas, like there's a small little window where the camera goes in, but they'd still like find it and just like immediately like jump on you, like heat-seeking missiles. And they're tiny. It's just, they're really good at like jumping and getting everywhere. And it was just kind of I was like, I really want to film these in action, but I also don't want to bring them home, and I don't want them to like infect the museum. And like we're working with one flea at a time, and I'm going to spend as much time as I need to on my hands and knees trying to find that flea that got out of the thing, because I'm not going to let fleas escape. And so I was glad when I captured some things that I could just say, okay, the adult flea work is done. Like we're moving on from that. So that was my least favorite.
How do you come up, I know we're jumping ahead a little bit, but how do you come up with what to look at next? Like, what is your next video subject? How do you decide something like that? What is the criteria for you? Is it run by the museum? Is it driven by you? And how do you gauge if it's going to be of interest, I guess, to the masses as well?
Yeah, it's up to me, and I have 2 rules I set for myself, and I've had in place for my content on YouTube for several years now. Everything that I make has to have these 2 rules in it. One, it has to be something I think is new. Like it has to be either footage of an insect that hasn't been filmed, or footage that looks different from what everybody else has filmed before, or it has to be a story no one's told before, or putting a person and their work on screen that hasn't been shown before. So everything has to have something that I feel is like new in it. And then number 2 is it has to be a reason for me to be the one presenting the footage and the story. So there has to have some sort of way I can relate myself or my interests or my professional sort of experience to. So that means I don't film. I try not to film things that a lot of other people that you can go on YouTube and find stories about. And it also means like I don't do kind of research stories that are far afield from kind of my area anymore. I would make a research story about, there's one on the channel, an older one about an astrophysics discovery of a person who works across the hall in astronomy. Like I don't make those videos anymore because of those 2 rules I have in place. And that's helpful for both the content on YouTube and like the story and like why I make the thing, why does the thing exist, but it also feeds into the research too, is that like a good proportion of the stuff on YouTube is are things that are research projects or will contribute to research projects, or things that I'm kind of just scoping out is like, hey, can I like film this thing to make an interesting public story, but also like make take it in the direction of being a contribution for some new piece of science. So like the flea thing, I spent most of my day today writing a manuscript about larval cat fleas, which is why we had the adult fleas in the lab. I had a student working on them. So that's a story we put out on YouTube, maybe like close to 2 years now. But that's now, an almost complete research manuscript too. So the 2 things kind of work in hand now.
That also, like, why did you have the fleas in the lab? Like, how does your lab work, I guess, too, right? So it's like, maybe it might be with your postdoc, right? But becoming like the Ant Lab on YouTube, right? And then you have the trap jaw ant videos, you've got the leaf cutter ant videos, then we see springtails hopping, and then there's now also, right, fleas. How do you choose what to work on?
Yeah, I mean, it's changed over the years, of course. I used to just exclusively work on the ants. Now we still have ants in the lab. I don't have any active ant research projects at the moment. And, now it's still centered around insect behavior, but it's broadened into kind of any insect that I feel like we can make a scientific contribution with. By working on, and at the same time, I try to make sure every project can produce interesting public-facing stories too. So these days, the goal of my research is really to, while making scientific contributions, is to give people a reason and a means to connect to and appreciate insect and arthropod biodiversity. And so I view kind of 50% of my job is research and teaching, and 50% of it is public, media making and thinking of ways to make insects and arthropods publicly engaging to people. Because I do feel like that's, something I enjoy doing and can do, at a certain level and have an interest and passion for it, so I should use it and devote a lot of my time as a scientist towards that.
It's definitely not easy, right? I know my wife and I, we do school visits and like senior centers, and we tried originally to show pictures and videos that we took of insects, and it was always met with this like ew feeling, like, I don't know, that's kind of gross. So our path was like making little cartoon characters of the insects that we've worked with, like, dressed as like Marvel superheroes or something. And that kind of lowered the guard of the kids. And then they were able to get past that fear and get into it. When you're saying like, this, like, I'm going to take a video, let's say of the springtails, and this is going to be exciting to people. How do you end up doing that gauging? Because that feels like in the science communication, like no one really teaches you how to do that. So like, how do you get that incredible feel of like, this is going to hit and people are going to find this interesting? I know for us, we find every one of your videos interesting, but we might be like the edge case, right? Because we're insect scientists.
I don't really. I make videos primarily for myself. I think I'm the most important audience member for my content, actually. Maybe people don't talk about it that way, but I think it's true. Is that when a goal of my content isn't to convince people that insects are interesting or to change anybody's mind or feelings about insects. That's never a goal of mine. What a goal is that they see the content and that they, at the end of the day, whether or not they think the thing is gross or cool or interesting, they have met someone and heard from someone who does think it's cool and interesting and valuable and worthwhile. I don't need them to change their minds about insects. I just need them to hear a perspective that like talks about insects and appreciating them and biodiversity and appreciating that on its own terms for its own sake. Not because it can, lead to a new technology or solve XYZ human challenges, but just because like it's worth it, it's valuable, and its own sort of, reasons for existing. And I want them to just meet a person who expresses that. I don't, whether they think it's cool or not, like, I want them to leave with like, oh, I watched this video of this guy who like studies these bugs in his backyard, and the bugs do these backflips, and he thinks it's really cool. Like that's it. That's the message, right? And so yeah, and that's why I kind of have those rules from my content. Is like there has to be a reason for me to think it's like worth making a story about and worth telling people about, and that's what I want to put in my content, rather than like let me give you the Wikipedia entries about why this thing is important. I want like a subjective kind of story about why I think it's important. That might be because it's useful, but more likely in my case, it's just because, gee whiz, this thing looks cool, doesn't it? That's it.
I mean, I think that's what attracted us to the channel anyway.
Yeah.
Because it seemed to be driven and inspired by curiosity. There isn't like the underlying tone of like how this might help human genetics down the line, which is fine. There's nothing wrong with having that.
Yeah.
But it's just like, oh wow, this is really cool. I'd never seen this before. Now I get to see in slow motion how they like push the water up and they don't break the surface tension. I never even thought I could see something like this. And it's just, I think the chat's echoing to just like how exciting it is to be able to see these things, and that whenever we watched your videos, that excitement on your behalf and your passion really, really come through. Like I love the video with the hoppers, and you're collecting them in the backyard,
Oh yeah.
and you're showing us how to do it, and it's just like, oh man, he really is excited about doing this and showing us how they can
Yeah.
fly. And I can't tell you, we've watched that video countless times because we find hoppers in ancient amber, and then we always refer to your video to show how they would jump.
Nice.
And it's like every single time, all right, ant lab video, let's go. And it's just, it's always so cool to be able to see it every time. Like it hasn't lost its gleam on your videos, and it's, you know, I love that passion shines through.
No, that's cool. That's great to hear. Yeah, awesome.
So so how did you end up, I guess, where you are today? So you finished up your graduate work. How did we get, you know, Dr. Smith working at North Carolina museum, right? Part videos, like had big on YouTube. Like how did we finish up getting to that stage?
Yeah, well, I did a period of postdoc work at University of Illinois. And at the time, I did that for, I think, 5 years, and I was applying for jobs. And in Illinois, I started like really making media as a postdoc. So the first kind of big public thing I did was I produced a podcast of where I would like go and do long form hour-long interviews with like kind of senior established scientists and ask them about like their career paths and their motivation. And these are people who like wrote books and stuff, and I read all their books and do a ton of research, and I would only do the interview if I could go and sit in their office with them. So I would like drive for hours and like, wow, go to Minneapolis and like, interview Marlene Zook or something like that. And, like got all the recording gear and like made it as like, legit as I could. And did that for a few years. It was a lot of work, but it kind of like taught me a little bit of audio media production and kind of, got me started on like, yeah, I'm just going to like make the media I want that I want to exist and just make it myself and put it out. And so like I took it seriously and I did it for a long time. And then I also, towards the end of that, started making videos and made them as for the purposes of press releases to accompany a press release. And so they were explainer videos about, scientific studies, either that I did, mostly from what I did, but also from colleagues. And so having that media making experience, I found a job ad here at the museum that wanted scientists who could do some form of public engagement. And so I applied for that job. Didn't really expect to get it, but I had and was at the time had like a legit resume with SciAnts, like a bunch of publications and things like that, and then all the media production stuff and the self-publishing sort of years of podcast production, but also videos, and that kind of got me in the door here. That's how I got the job, where there was, they wanted to bring in someone who was doing public engagement, and luckily I landed here, and it kind of gave me the space to devote, a significant portion of my time into developing media production. And, then I stopped doing the podcast. I went full into video making and sort of, it was slow kind of getting good at that and figuring out what to make and why to make it and what to say and what I should be concentrating my time on, or even how to do YouTube. That took me a long time to figure out, like, to use YouTube in a way that is not just for video hosting, and to figure out that, like, oh, what is a YouTube channel? I'm like, what should this be? That took me way longer than it should have. But yeah, and so now I've kind of narrowed it down to I don't do that many things. I just make media. Like I say no to a lot of things. And I don't write blogs. I kind of like concentrate my time in media production and research, and kind of that's it. Because I don't know. It takes me a while to make every YouTube video I put out, for sure.
I imagine that's, like you said, hours worth of recordings just of like the flight, let's say, and if they're free flying in the lab and you have to like get them back onto that little square that we were doing the recordings, like I can't imagine the number of outtakes, right? And just like the day-to-day upkeep of the insects, even there just seems like there's a lot of background work that goes into it. With during your postdoc, it sounds like maybe that the professor was supportive of you being able to pursue this passion as well.
Oh yeah, for sure. Yeah, with the postdoc, that was my advisor Andy Suarez that I worked with. Was totally supportive of me at the time producing that podcast. Would even recommend people for me to go interview. So yeah, he was totally game for that. That's when I started working with trapjaw ants again, because he had worked on Odonomachus, and I wanted the trapjaw ant genus, and I wanted to get back into that and sort of go back to my roots with interest in ants. And so went back to Florida for field work and brought them back to Illinois and worked on them with him.
So it came full circle.
Yeah, for sure. Mm-hmm.
What was what was your research question at the time, and what was your favorite or most exciting result from that work?
Yeah, one of my goals for going back with trap giants was to repeat a little undergrad project that, at the time I didn't really know how to do science, but I was just messing around with the ants. And there's one ant in Florida. The species it's a Formica species, Formica archboldi, that co-occurs with trapjaw ants, and has this weird thing. If you encounter it in its nest in Florida, like they're littered with the head capsules of dead trapjaw ants. And so I was trying to, as an undergrad, work on their potential prey preference for them. And I did a little thing and presented a poster at a conference, but then it wasn't good enough to publish or anything like that. So as a postdoc, I went back and revisited them. I should do my 19-year-old self a solid. And now that I know how to do science, finish this project. And so I did, and that was a cool little project, and it was a cool story showing how these Formica archboldi are really adept at spraying trapjaw ants with their formic acid, and trapjaw ants in this genus are really susceptible to that and will kind of immediately keel over and die if they get splashed with this archboldi's spray, and they do indeed bring them back to their nest and eat them and keep the bones in their nest. And so it was a cool little study, and I at the time was making videos, so I made a video press release about it and dubbed it dubbed the Formica Florida's skull collecting ants. And that story did really well because I had a bunch of cool footage, and who doesn't want to hear about a skull collecting ant from Florida? It's just a fun thing,
Is that also on your Ant Lab YouTube page, or is that located elsewhere for the folks interested?
Yeah, that's a video on the Ant Lab channel. It's an old one. I think at the time I was making a form of video that was really popular was short words on screen, videos that you could watch on Facebook without sound on. So it was one of those style videos.
Gotcha.
So it's way different from what videos look like now on my channel. But just you have to transport yourself back in time, 8-10 years ago or whatever.
It's really cool that you got to go back, like you said, to your 19-year-old self and finish out that research question. I feel like a lot of folks, right, is if we start off in research and we see something really cool, usually there's not a resolution, right? It's not even necessarily picked up by someone, but it's really cool that you had that and it turned into a paper. I have a bad habit of picking really dumb goals that I want to achieve in science. And that was one of them.
One of my other goals was to publish on something from my backyard. And so all the springtails in my springtail work are only collected from my yard at home.
How much of a yard are we talking? Is there a forest, or is it just a regular size yard?
Yeah, we're in Raleigh. I'm not very far from downtown. It's just a regular house and regular yard.
So really accessible to anyone.
Yeah, for sure.
That's really cool.
Mm-hmm.
We had a question from the chat is, how easy was it to transport these ants from Florida to Illinois? How is the permitting for something like that work?
Oh, yeah. Well, you have to get USDA APHIS permits to interstate transport the ants, but also to house the ants alive in their destination. So at the time in Illinois, we had a quarantine facility for collecting out of state ants, but also out of country ants, and bringing them and working on them.
Did you have, were there any other insects in the lab that you really enjoyed studying? Was it primarily just the trap jaw ants at the time?
Yeah, at the time it was just the trap jaw ants. We had ant species called, for a brief amount of time, called Bothroponera, which are foaming ants. They would like foam their venom, which is pretty wild. We did a little study. I'm not sure if that's out or not yet, but that was pretty cool. Foaming ants. Just for that weird little venom foaming thing that they do. But yeah, for me, it was mostly just the trap jaw ants.
It's really cool. And we actually got 2 giant raids from our friends Creatrix Britt and Melissa with all the numbers. Raiders, welcome in. My name is Balint. I'm 1/3 now of the SciAnts stream crew. And tonight we're joined by Dr. Adrian Smith, who is an incredible researcher, science communicator, specializing on insects. And we're asking him and talking about his research. He runs a YouTube page called The Ant Lab that's linked at the top of the chat. Raiders, everyone, welcome on, and thank you all for bringing over your communities. Feel free to put questions into the chat. We've got Dr. Smith for about another 10 minutes before he has to go, because he's also East Coast, and we don't want to keep him up to the hours that we stay up here on the stream. Thank you all very much for the raids. Everybody, welcome on in. So now that you're at the museum, we had a question from the chat, is there so far an organism in which on the video side of things that you've been able to work on that was kind of a dream come true video to put out? And then is there something that you would love to record? Like if you had access to all the bugs on the planet and I could just magically make them appear, what would you want to record?
Oh, I mean, tons of stuff. You have a huge list to record things, and I occasionally check things off. I mean, the last video I put out earlier this week of rogue beetles folding and unfolding their wings was on my list for a long time, and I tried to film that with other species until I found the right one that would do it. But things that I've checked off my list are like earwig wing folding was on my list for a long time. One video that I really love is the batfly video on the channel. Batflies are ectoparasitic wingless flies, like houseflies, flies that live in the fur of bats and feed off bat blood. And I'm lucky enough to work here at this museum where there's mammalogists, and one of our mammalogists goes out and does bat censuses where you mist net and collect bats and give them tags and release them. And so I went out with her a couple trips until we found the right species of bat with bat flies, and was able to collect one of these bat flies off of a bat and bring it back to the lab. And only lived for like 10 hours off of the bat, but was able to film it running around doing behaviors in the lab. And so that video I particularly love, because it's like, where else are you going to find footage like that? Like all the stars have to align for that footage to exist. Like,
yeah,
there has to be someone with a high-speed camera who knows someone who works on bats, who has permits to touch bats, and the right species that has the bat fly and that lives close enough to the lab where they can bring it back and film it. And like all that footage just won't exist again.
I mean, it kind of goes back to your mission of choosing what to record too, right? That you want to make sure that you're able to record things that, it's special to you, it's unique to you. And that's definitely one. Like I said, it's only 10 hours, so as soon as you catch it, and misting also isn't easy, because if you get nicked in any way or a scratch, you got to go get rabies treatments. And
I wasn't allowed to touch the bats, like, because you need like rabies shots to even work with them. All the conditions just had to be in place for that footage to exist. And then the cool part of that too is, like, I put it out, and in the video I was like, I would just talk about, like, man, look how mind-blowing this thing is. And one of the things that was wild is that they're totally wingless, but shockingly in the footage, when I started looking at it, you saw the hull tears, the little gyroscopes still on the batfly, and weirdly enough, they flap, they move when the bat fly is like running around. And I was like, this is wild. Like, I didn't even know they had hull tears.
Yeah.
And they still flap. Like, and I say it in the video, I was like, I can't believe this. This is crazy. And I put the video out, and then a week later, I got an email from a researcher at Case Western University who studies fly halteres, basically. And they were writing a review paper about halteres being used in non-flight scenarios, and they emailed me and was like, I think you're the first person to ever record like a truly wingless fly with flapping halteres. Like, can we use this footage in our research paper? I'm like, that's it. That's the entire spectrum of the reason why I make YouTube content.
That is incredible.
Yeah.
What a cool email too to then be able to get like, you are the absolute first. How can we showcase this? Like, that must have also felt surreal.
Yeah, like it's just like that's like the reason. Like, well, that's one of the reasons. It's like I was pretty sure this was like weird and like interesting and like unusual, and to like, you know, get that email afterwards and be like, oh yeah, can we like use this in our paper? Like, well, that's it. Right, that's the whole, that's, you know, a big portion of why.
What's just a fun little behind the scenes for you, our moderators in the chat. I've already found the skull collecting video and the batfly, and we have it ready to go to watch after we say goodbye to show off to the community, because it's really cool to hear these behind the scenes stories too, and just like, again, how your passion and curiosity are just shining through. It's just it's really, really exciting again, and thank you so much for joining us today. Like, I know you're very, very busy, and to take time to come to our Twitch stream and, you know, share your love of science is really, really appreciated.
Oh, I'm glad to do it. Yeah, thanks for having me.
As we wrap up, because I want to be mindful of your time, is there anything that you'd like to leave us with in terms of, like, I guess, what's the next big insect that you're looking forward to recording? And then if you have any message to people about just, you know, I guess, you know, honestly doing science on their own and just getting excited about it.
Yeah, I mean, you know, like I said, the type of science I do is all about trying to see and appreciate the world in a new way. And that results, for me, it results in YouTube videos and it results in, you know, scientific papers, but they're both doing science. They're both, you know, trying to make observations and contextualize them and like learn from them, you know, using the scientific method or not, you know. And so that's something I think anybody can really do. I mean, it's, you know, I'm lucky enough to be in a position where I get paid to do it, which I'm very fortunate for that, to have a job in science, and fortunate to have access to tools that help me see and appreciate the world in a new way, like, you know, slow motion cameras. But, you know, you don't necessarily need those things to do that, to do that type of science. And so, yeah, the stories I'm excited for are, you know, there's always, you know, I mean, currently there's like, you know, gigabytes of footage on here that like have yet to make it onto YouTube. And how it works is I pretty much always am recording things, and then it's just a matter of like figuring out what the story is and how to package that and like what to say about it. So there's tons of new stuff on here. And then, you know, we're doing really some of the footage that we recorded for research purposes is like some of the coolest footage I've ever seen. We have a student of mine who was working on jumping plant lice. I put out a YouTube video of plant lice a while ago. It's a really good video, but didn't get that many views. But they have spring-loaded hind legs, and they do the fastest front flip on Earth. And we're doing some descriptive work with them, but also some predator-prey interactions, because these jumps and the jumps of, you know, many things like hoppers, like springtails, are supposedly for predator avoidance, but no one actually films that part. No one actually films them using the jumps and using their high-speed things to actually escape predators. And so we've been doing a lot of that with plant lice, and like, man, the stuff we're getting is like awesome. It's really cool. But that won't be out for a while, because we're gonna you know, get do the work for the science, and then, you know, as the science wraps up, we'll start making the public facing stuff. But I can't wait to debut those stories, because they're really cool.
That sounds really exciting. Are you using the real predators as well, not like some a proxy?
Yeah.
Nice. Yeah. Oh, that's gonna be really exciting.
You have to guess what the predators are, but we found the perfect predator, which is the perfect type of predator. We found the predator who, in the lab scenario, as it touches one of these lice, one of these psyllids, it is unmistakable for an act of predation. There's some things that like can run around and just bump into a thing, and you're like, oh, did it bump into it because it wants to eat it, or is it just like running around willy-nilly and just touching a thing? We found a system to use where it's like unmistakable that this thing is trying to eat this thing, and this thing has, a couple nanoseconds to escape or die. So
I can't wait to see that.
Yeah.
That is a perfect teaser. As we sign off, I know we have plugged we're plugging your YouTube page as well as the flipbook. Greg came in and dropped in the flipbook link. We didn't have it before, but is there anything else that you have upcoming that you want to promote, where to find you, or anything at all that you would like to plug?
Oh no, just the YouTube channel, the Instagram, publish shorts there too, and I wish I could make more stories all the time, but they will trickle out slowly, and hopefully there'll be more cool stuff up there soon.
Awesome. We will link the Instagram as well and have it pinned to the top of the chat so everyone can go follow you. And guys, blow up those likes, the comments, and we're gonna watch some of your videos now too that you were talking about and pulling back the curtain. So thank you, Adrian, so much for your time, your expertise, and honestly just sharing your passion with us. It was really exciting and really refreshing too to see.
Awesome. Well, thanks for having me. I enjoyed it.
Thank you so much. Till next time.
All right. See ya.
Bye.